Welcome everybody, and welcome all our viewers online. Before I introduce Dr. Breining, I want to remind you that in 2 weeks' time we have another guest speaker here, Adam Buchwald, who will be speaking about motor speech production. And it's important to know that motor speech production, but not phonological production, is improving or improves after motor learning training. So we'll be looking forward to that one. Again, if people want to look at old C-STAR lectures, you can view those on our YouTube channel. The address is right on your screen right now, and you can also go to our C-STAR website where you can look at past lectures and also find out more information about the Center for the Study of Aphasia Recovery. It's a pleasure for me today to introduce Dr. Breining. Bonnie Breining is a postdoctoral researcher at Johns Hopkins University in the SCORE lab. She was educated at Rice University and the University of London before getting her PhD at Johns Hopkins University. And her doctoral research centered around semantic and lexical item characteristics, the effects of those characteristics on learning outcomes in acquired dysgraphia. In general, her research centers around cognitive mechanisms that underlie both written and spoken language, and currently in the C-STAR project she's involved with studies looking at effects of transcranial direct current stimulation to improve treatment outcomes in acute and subacute aphasia. Today she'll be talking about action and object naming, long-standing discussion, so we'll be looking forward to her findings in acute stroke and also in primary progressive aphasia. So Bonnie, the floor is yours.
Thanks very much. So as Dirk just said, I'm going to talk about deficits in object and action naming with evidence from acute stroke and primary progressive aphasia. So after stroke, it's very common to give picture naming assessments in order to test language outcomes. This is good. It taps a lot of the components of the cognitive architecture of language. So you have to go from seeing a picture to producing a word, which involves recognizing what the picture with object recognition mechanisms. You have to understand the semantic information about it. Then you have to be able to retrieve the appropriate word form from long-term memory and then hold the segments of that in working memory while you're producing it. So it's a nice test to tap a lot of different components. For example, the Boston naming test is very frequently given, which would be showing people a bunch of black and white pictures, ask them to name it. It's a nice test because it elicits a range of performance among both healthy controls and also among patients. Although for the items that people do know, there's really good name agreement. However, many of these assessments that we commonly give are focused on objects when neglect actions. It's really important to realize that you need action naming too. In stroke, a lot of selective deficits for objects than actions have been observed. More frequently, people are okay on objects but have problems with actions. However, the opposite has been observed as well. It's not just that actions are more difficult to name than objects are. And so because we have these kinds of selective deficits, if you only give object naming assessments, you may be insensitive to detect certain deficits, and then you'll be unable to actually understand the patterns of recovery. Action naming is important in and of itself in that you are going to have a lot of trouble producing sentences if you can't retrieve verbs. So it taps some knowledge that is necessary for communication. It's also important to note that the dissociations between object and action naming are not limited to stroke. They're also found in other etiologies, including primary progressive aphasia. Now, I know a lot of you all who are involved in C-STAR spend most of your time thinking about stroke and much less about primary progressive aphasia, so I thought I would just say a couple things about it. So primary progressive aphasia is a neurodegenerative condition that involves increasing language deficits for 2 or more years in the face of relatively intact cognitive function in other domains, such as episodic memory, nonverbal memory, and visuospatial processing. So those are like the general characteristics to be diagnosed with primary progressive aphasia. And then most people are subtypes into one of three variants. There's the semantic variant, the non-fluent or agrammatic variant, and the logopenic variant. There's also a group of people who are unclassifiable because they present just with anomia and dysgraphia, which could be any of these variants. But many of those eventually progress to being classifiable under the system. To give you a little information about each of them, the semantic variant is diagnosed on the basis of impaired naming and impaired single word comprehension, which may also involve impaired object knowledge, especially for things that are low frequency or low familiarity. People with this variant also often present with surface dyslexia or dysgraphia. In these participants, there's often atrophy in the anterior temporal lobes, predominantly on the left side, but bilaterally. And their pathology is often frontal temporal lobe dementia. For the non-fluent agrammatic variant, these people have either agrammatic production or apraxia of speech, and they often have impaired comprehension of syntactically complex sentences, although their single word comprehension is relatively spared. Damage for them is generally in the left posterior frontal insular regions, and again, they have frontotemporal lobe pathology. And finally, there's the logopenic variant. These people have basically deficits in phonological short-term memory. They have difficulty with repetition and with single word retrieval. They will make a lot of phonological errors in spontaneous speech and naming, but they're not agrammatic and they don't have apraxia. Their damage tends to be in the left posterior perisylvian or parietal regions, and their pathology is the same as happens with Alzheimer's disease. Important for our topics today, it's been found on a number of occasions that semantic variant patients tend to have disproportionate difficulty with actions, whereas non-fluent ones have disproportionate difficulty with actions. And so we see that there's behavioral dissociation in both stroke and PPA between objects and actions. This indicates that there are several neural substrates for these types of naming that can be damaged independently. So we not only have this converging evidence from stroke and PPA, but we also have evidence from functional neuroimaging of neurologically intact adults that goes along with this. You can find different regions that are involved when people are naming nouns versus naming verbs or object fractions. In general, the findings can be summarized as that there are two distinct networks for actions. These involve left posterior frontal regions, whereas for objects, they involve left temporal regions. However, there are some inconsistencies for different groups. For example, in the PPA literature, there's a whole lot about the left anterior temporal pole's role in the representation of objects, but we very rarely find that people who have difficulty naming objects after stroke have damage in this area, partially because you don't really have damage here unless you have a very large stroke. Therefore, it can be interesting to look at these different groups and compare what you can get. So in this talk, I'm going to talk about some projects that address 4 hypotheses. The first is just that some individuals with aphasia have impaired action naming. This is very likely true based on the basis of all this. And so to look at this, we developed the Hopkins action naming assessment. We're hoping that it was a sensitive enough test to detect these kinds of effects. The second hypothesis is that there's a double dissociation in performance for object and action naming. And to look at this, we can compare performance on the HANA that we developed to the test of object naming, the Boston naming test. Assuming that we do find differences in behavior for these, we could also look to see if there are different neural substrates implicated in object versus action naming. And to look at this, we used an atlas-based analysis to compare regions that are associated with the different naming tasks. Finally, the hypothesis is that comparing individuals with aphasia due to primary progressive aphasia and acute stroke will provide a more comprehensive picture of the brain-behavior relationships critical for naming. This could take 2 different forms. One is that we could find really similar patterns when people are tested on the same tests, regardless of their etiology. The other is that we might get complementary information from the different etiologies because they have different kinds of damage. And so to look at this, we included participants with both types of aphasia in our study. So just to be able to evaluate action naming deficits, we developed an action naming assessment. When we were putting this together, we realized that we were already giving all of our patients the 30-item Boston naming test. It's a pretty standard test. It goes from more common, frequent items like bed that everyone gets right to less frequent, more difficult ones that even healthy controls often miss, like trellis or protractor. And so we wanted a comparable action naming assessment that would have a range of performance and be matched on frequency and length to the Boston test. So to do this, we took a large pool of pictures of actions and had them normed by 20 young controls. We got 35 items that had at least 75% name agreement. From this, we further narrowed it to 30 items to be matched to the BNT based on the performance of 26 older adult controls who were 40 to 83 years old and very well matched demographically to our PPA participants. Many of them are actually spouses and caregivers for those patients. In this test, we didn't specifically control argument structure. Every time I talk about this, someone asks. But it is true that there is a mix of transitive and intransitive verbs. So 6 of them were, of the 30 are intransitive, 17 are transitive, and 17 can be used either transitively or intransitively. So these are the items that we picked. They go from relatively easy to name ones, like run, to much more difficult ones, like hypnotize, tapping much less frequent. And so we were able to, from these control data, determine what would count as normal performance. We see that in general, people do seem to do a little bit better with the objects and actions. On average, they got 26.3 right on the BNT, but only 23.9 on the HANA. And there was a larger range of performance on the HANA. From these, we developed cutoffs that were 1.5 standard deviations from performance of the normals, which would give us, you'd have to get at least 23 items on the BNT or at least 19 items on the HANA to be considered intact. Anything less than that would be impaired. Then we were able to give both the BNT and the HANA to the individuals with aphasia. We gave it to 138 different individuals with PPA and to 37 who were with acute stroke. So they were within 48 hours of an acute left hemisphere ischemic stroke. We actually have given it to quite a few other stroke patients, but some of them have things like bilateral damage or hemorrhagic stroke or slightly outside the range. But 37 of them fit our criteria perfectly. So this is the performance of behavior for the primary progressive aphasia patients. On the x-axis, you have their score on the HANA. On the y-axis, you have their score on the Boston naming test. And then these lines are the cutoff points. So of our 138 participants, 33 of them had semantic variant, 41 had non-fluent variant, 50 had logopenic variant, and 14 were unclassifiable at the time of test. And as we had hypothesized, some of them were in fact impaired on action naming as shown by the performance on the HANA being below the cutoff, so all of these people. There were also some that were impaired on Boston, all of these people down here. 96 of them, in fact. You'll notice that for most people, they're either impaired on both or intact on both. But we do have some dissociations where people are better at naming objects and actions or vice versa. So these people are intact on the object naming but impaired on action naming according to the cutoffs. And these people show the reverse pattern. We also did some direct comparisons of scores on the BNT and the HANA using Fisher's exact tests, and we found that when directly comparing their scores, 2 people scored significantly better on the HANA than the BNT. They both happened to be semantic variant patients, and 11 people did better on the BNT than the HANA. They were a mix of variants. Also, in accord with past results, the semantic variant patients had a bigger disadvantage for objects than actions relative to those who had the non-fluent variant. So this is the same chart, but for the acute stroke patients instead of the primary progressive aphasia. Here there are 37 of them, so you see it's fewer things on this chart. As hypothesized, we do again have some who are impaired on action naming, so all of these over here, and some who are impaired on the BNT. And just like before, there's a double dissociation between the objects and actions. There are some people who are okay at naming the verbs relative to control performance, but are not okay naming the nouns. But and then there's one here who is in fact better at, who is intact on the object naming but impaired on the action naming. And again, we can directly compare the BNT and the HANA scores, and we find that these participants who I circled in red and blue had significant differences in their scores, so they did much better on one than the other. So this person circled in blue actually got 27 of the HANA correct, which is pretty high performance even for controls, but was clearly impaired on the Boston. Whereas this person in the red only got 3 right on the HANA, but got 12 right on the BNT, which is different performance. Since we found a behavioral dissociation, we can then look at what the neuroimaging shows us about which areas are related to performance on these object and action naming tests. So out of the 138 PPA patients who I showed you your behavioral scores for, 31 of them had scans within 6 months of assessment. 6 of them were semantic variant, 12 non-fluent, 11 logopenic, and 1 unclassifiable. And those same 37 acute stroke patients were imaged within 48 hours of stroke. I then performed an atlas-based analysis to investigate the neural substrates associated with the object and action naming. This used MRI Cloud, which is an online program that was developed by some of the radiologists at Hopkins. What it basically does is it takes the original brain of the participant that you give it. It does some reversible transformations to take it to the same space as an atlas. The atlas has been parcellated into 283 different anatomical regions, which can then be applied to that normalized brain and then converted backwards into the native space of the participant and applied to the brain, so that you end up getting measures of the volume of each of these 283 different areas for the brain that you gave it. So in this study, we chose not to look at all 283 of those regions, but to focus on 20 that have previously been reported to be involved in naming and are likely to be damaged in at least some of our participants. These included frontal areas like the left inferior frontal gyrus, the pars opercularis, pars orbitalis, and pars triangularis, pre- and post-central gyrus regions, the left supramarginal gyrus, the left angular gyrus, and the left insula, and then temporal regions, both left and right hemispheres. So the superior temporal gyrus, the superior temporal pole, the middle temporal gyrus, the middle temporal pole, the inferior temporal gyrus, and the fusiform. In the analysis of primary progressive aphasia patients, we had to do some things to make it interpretable. So neurodegeneration in PPA is not straightforward to delineate. It's not like a stroke where you can see the lesion and just trace it and then go from there. You can see that there is some kind of shrinkage, but it's hard to say exactly where it is and what size it would have been before. So instead of looking at the amount of damage in each region, we used the measurement of volume in that region controlled for regional atrophy, which would be the volume in the region divided by their intercerebral volume. Then to analyze this, we did a regression analysis where we predicted the behavioral score from the volumes in each of our regions of interest. And to control for differences in brain size, we added a ratio of intercerebral volume to intracranial volume. What did we find? For the primary progressive aphasia patients, there were associations for their scores on the HANA. so action naming. So these people had volume was related to performance in the left inferior frontal gyrus, the pars opercularis, which goes with a lot of other things that people have reported for actions being in the frontal regions. They also showed relationships where degeneration in the right superior temporal gyrus, left middle temporal pole, and right inferior temporal gyrus were related to poor performance on these tasks. For their relationships with the Boston naming test of objects, again, they came out to be right superior temporal gyrus, left middle temporal pole, and right inferior temporal gyrus, not the frontal areas. And also brain size seemed to matter here. For the stroke, we are able to delineate the specific lesions that people have. We were able to trace the lesion from their acute diffusion-weighted scans, co-register that with high-resolution anatomical images, and then before we segmented those, we dealt with the fact that there was damage by flipping tissue from the undamaged homologue, so the right hemisphere, to replace the lesion tissue, and then we can segment from there. After we get the segmentation, we can measure the amount of lesion in the region by overlaying the lesion onto the segmented brain. And the measure that we then can use is the percent of damage to that region, which is the volume of the lesion in the region of interest divided by the total volume of the region of interest. Again, we did a regression analysis to predict the behavioral score from the volumes. This time, instead of controlling for brain size, we controlled for total lesion volume, which is very often related to performance. In this analysis, we also necessarily had to reduce the regions of interest. When you, as I'm sure you will all be shocked to hear, when you pick out left hemisphere patients, they don't have damage to the right hemisphere, so you can't look at any of those areas. It also happened that in our sample, no one had damage to the left middle temporal pole. So we didn't look at that region either. So that removed 7 of our 20 regions before analysis. For the stroke patient, damage was related to lesion volume, volume in the left angular gyrus, and the left fusiform gyrus. These were what were related to the action naming. And for the object naming, there was damage to the left angular gyrus, again, but not damage to the fusiform. Instead, there was damage to the left middle temporal gyrus as a predictor. So these are the results combined. So you can see that you get different regions for the PPA and for the acute stroke, but that within an etiology, there's a lot of overlap in the regions that are involved in the processing of objects versus actions, but there are also some differences. So as we hypothesized, there are several neural substrates for object and action naming. For primary progressive aphasia, damage to the left inferior frontal gyrus, pars opercularis, was only related to naming of actions, whereas for stroke, we found the left fusiform only for actions and the left middle temporal gyrus only for objects. However, within an etiology, the overlap of regions, there was overlap of regions involved in object and action naming. So most of the people who are impaired on one task were also impaired on the other. That might be one reason for this. Additionally, there are very similar cognitive processes involved. They might be doing really the same thing for some components of the naming. It could be that our regions are really a little bit too big, that our scale isn't small enough to see where the differences actually are. And then there's the practical problem that when you name pictures of actions, it requires you to recognize objects. It's almost impossible to depict actions in static 2D pictures without showing some kind of object. And so when you're recognizing these pictures of actions, you're also doing object recognition, which would involve exactly the same processes and so should show the same areas. It's also important to note that we did identify different regions in the analyses of primary progressive aphasia and acute stroke, showing that we get complementary information by looking at more than one type of participant, when the differences that we find are likely related to differences in the regions that are damaged. For instance, the ones that we found for primary progressive aphasia, almost none of them were actually regions of interest in the stroke analysis. They just don't have damage there, so it would have been impossible to find them. So another thing we can look at in the future are some longitudinal analyses. So far, what I've been telling you about are analyses about a single point in time, but change over time can also be informative. So in primary progressive aphasia, people continue to get worse, which is unfortunate for them, but does allow us the opportunity to look at which areas of atrophy are related to their decline. And also in stroke, on the other hand, people get better. So we can see if there are areas that are critical for recovery. So for instance, if you have damage to that region, do you not recover? Or is damage to that region something that you can overcome in the future? So I have a little bit of preliminary data to talk about with these things. So we can talk about atrophy and decline in primary progressive aphasia. So this is a scan from MJE, who was a participant with semantic variant primary progressive aphasia who showed worse performance on objects than actions. This is his first scan. And then this is another scan of the same participant 26 months later. Using similar tools to the allosteric analysis, we were able to transform the second scan to the same space as the first, and then we can look at local volume change. So that's what this map down here is. Areas that are more on the red end of the spectrum, so like oranges and yellows, are regions that expanded, and regions that are more on the purples and blues, those are regions of shrinkage. So what we found was that there was shrinkage in the anterior temporal and nearby frontal regions. It was very focal in this participant who was bad at the objects, which suggests that perhaps those anterior temporal regions are really important for the processing of objects. We also looked at a couple other participants with PPA who did worse on actions than objects, so the opposite side of that. So we have this participant who was scanned 2 times 13 months apart, this one was scanned 19 months apart, this one 27 months, and this one 22 months. For these people, you can see that there's more diffuse atrophy. It's not focally localized to the anterior temporal lobes like it was for that other patient. In fact, what you're seeing in a lot of these, like this person particularly, is a lot of expansion. So the ventricles have been getting bigger because the tissue around them has been atrophied, and so they're filling up with CSF. And one of the things we could do in the future is correlate volumetric changes with performance changes. So we could look at how these people did on specific tasks instead of looking at their overall performance and see how those relate to their volumetric changes. Another thing we're looking at is stroke recovery. So this project actually is something that a visiting medical student in our lab did this winter. He wanted to look at the regions that are critical for recovery and object naming. So he took 19 participants who had been assessed using the Boston naming test, both acutely, so within 48 hours after stroke, and then at follow-up 4 to 7 months later. And he found that although none of the regions that of interest to be looked at were predictive of their performance acutely, they were predictive of how well they did 4 to 7 months later. So those who had damage to the left posterior superior temporal gyrus or the left superior longitudinal fasciculus performed worse at follow-up than those who didn't have that. So it seems like those regions are, if you don't have intact tissue there, you have difficulty with recovery. And then in the future, we can do a similar analysis with the HANA and see how that relates to action naming instead of just object naming. And it also shows that the importance of looking at white matter tracks as well. So one of the best predictors here was the SLF, which connects a lot of the language areas. This is something that we should probably consider analyzing the rest of our data. So in conclusion, I looked at 4 different hypotheses. The first was that some individuals with aphasia would have impaired action naming. To look at this, I developed the HANA, and we found that it was, in fact, sensitive to this kind of deficit. So we were able to find people who had impaired action naming in both primary progressive aphasia and acute stroke. The second hypothesis is that there is a behavioral double dissociation in performance for object and action naming. So while the majority of individuals who were impaired on either the BNT or the HANA were also impaired on the other assessment, and those who were intact, the same thing, we did find a double dissociation among a few of the participants so that they would be better on one than the other. Based on this, we were able to see that different neural substrates are implicated in object versus action naming. So within each etiology, within primary progressive aphasia or acute stroke, there was overlap of the regions involved in both naming tasks, which allowed us to identify different regions for the objects in action naming. And finally, we did end up getting more information by including individuals with aphasia due to both PPA and acute stroke than we did from anyone alone. They provided complementary information, which led to identification of neural regions that would have been missed if we'd only looked at one. These are clinically important results, so we need to remember that we should assess not only object naming, but also action naming, and to consider both types of naming when developing treatments. And then future work looking at longitudinal change may add additional insight. And that's all I have to say. Thank you. Does anyone in here have any questions? No. We don't have any questions here. Oh, never mind. So she asked how far in onset were the PPA patients? Were they really severe? Were they pretty mild? There was a range. For the ones who had, so every time these people came in to get the same battery, for the ones who had come in multiple times, I tended to pick the middle one. So to try not to have the effect where they weren't missing anything, or the one where they couldn't name anything at all. So that seemed most likely to find the dissociations of interest.
Do you hear me? It's Tamara talking.
I hear you.
Okay, so I was wondering when you compared the primary progressive people to those with stroke, only in the acute phase, only after 48 hours. So why did you pick that time, not the chronic phase? Or?
Well, you can learn a lot of interesting things from looking at patients in the acute phase because they haven't had any time to adapt to their injuries. You're not seeing what's happened after they've recovered and they've started recruiting other brain areas. You can see more directly where the injury is having a real impact. Does that make sense?
So, okay. Because I don't know, because there is the, okay, maybe that makes sense if you just didn't make any.
You can learn different additional interesting things from the chronic phase as well, like whether those regions are necessary for recovering. But if you look at it before there's been reorganization, you can say what the normal organization was more likely to be like.
Okay. Thank you.
Oh, excellent. So I'm not familiar with this HANA battery that you were mentioning, and I was curious. So the pictures that you used to illustrate actions, were they mostly like human figures doing stuff?
Mostly.
Yeah. So, like, for example, you know, like this hypnotist, right? It's like it's a picture of a face, basically, right? So, and I was just curious, like, whether that could go, you know, like, there. I'm curious if that has anything to do with the fusion form that you see. So it could be the four-base area.
Oh, well, a lot. I think that the majority. I think you need to mute something over there. Okay. So human figures don't actually have faces in the HANA. There are a lot of pictures where they're hands, so like one of the items, so it's two hands. Some of them do have faces, like the Running Man is the whole man. But it is possible that it could have something to do with recognition of the human figures, since in the object naming test, I don't think any of the items in the Boston naming test have faces in them. No, we all think not. So you're right, it could have something to do with that.
And it's not possible that you guys can run fMRI on those, or on a similar set of subjects, right? To see actually which parts of the brain are activated by those stimuli. It's probably...
We have not done that. It's something someone could do, but we haven't.
Okay, okay. All right. All right, thank you.
Bonnie, I have another question. In the stimulus list, have you done an analysis of the items on the Boston naming test, but also on your action naming items as to, for example, concreteness or the action relatedness of the objects? So, for example, scissors are very much related to an action, whereas house isn't. Both of them are objects. So in some other studies, people have actually looked at more on a gradient scale, right? So to what extent is an item actually action related, rather than is it a verb or is it a noun? Do you have any thoughts on that? Or?
We haven't tried that, but I think it would be interesting. So like the ones in the object naming at task are very high in concreteness, but some of the, and we attempted to have the actions be fairly concrete, but some of them aren't the most concrete looking things. And so it probably would be something to consider. That also might be one of the reasons that even normal controls are worse on the objects than the action.
Right. And it's been a kind of pervasive question, I think, in this field, right? What is it exactly that accounts for categorical differences, if there are any? So
right.
it will be important to take a look at all the dimensions, but it's really hard to control for all these things if you have different data sets, I know.
Yeah, we had to go with the easy thing and just do it.
Just a different question I had on the double dissociation that you noted in the behavioral data, right? You basically said we consider someone to be impaired on action naming or object naming if they're one standard deviation below the score for your control.
1.5, but yeah.
1.5, yeah.
It was not the lowest cutoff.
Right, but that's important, right? Because you're making a strong, the claim of a double dissociation is a really strong claim that has a lot, has a lot of implications for the field.
Right. So we do also find it not just looking at impaired versus intact, but directly comparing the scores. And so found people who were much better on one than the other, which is less common than just being impaired on one versus the other, since many people are pretty close in their scores, but might be close to the line on one of them. But for instance, here we have these people who are much better at one versus the other. But yes, it is a very strong claim.
But it seems to me that that latter analysis, where you actually do a statistical analysis of whether someone is worse in one category than the other, that might be stronger than the claim that is based on being a standard deviation.
I think you're absolutely correct.
Any other questions from us? I think you have a question on the chat, Bonnie.
Okay, got to get back to it. Okay, so Kirana asks if I could comment on the right inferior temporal areas in primary progressive aphasia. I wonder what she, in that maybe she was saying that it's unusual to find right hemisphere damage to go with more of the language tests. And so, for verbs. Karen, I still don't understand what you mean. Where I was going to go with that was that it's unusual to find right hemisphere damage associated with a lot of language things, But in PPA, a lot of times people have bilateral damage. And what really might be happening is in order to see the behavioral effects, you have to be impaired on both sides. You have to have bilateral damage. You might not see that effect with just unilateral damage. That's a lot of the arguments that people have made about anterior temporal lobe damage. And it could be that something similar is going on, that you're not seeing the effects until they really have advanced atrophy that has affected both sides, and they can't use one side to compensate for the damage to the other. Oh, good. She meant what I said. Good.
No other questions here?
Anyone else?
All right. It doesn't seem that we have more questions from our end nor from the chat box. So thank you very much, Bonnie. That was great. Thanks a lot.